Hello students, welcome to EPG Pathshala. I am Dr. Nilofer from DPAS DRDO Delhi. Today I am going to talk about thermodynamics and kinetic approaches to membrane potential, calculation of electrochemical potential by Nernst equation from the paper Membrane Biophysics. And the objective of the talk are electrochemical potential, establishment of membrane potential, calculation of electrochemical potential by Nernst equation, physiological significance of membrane potential, and the transport across the cell membrane, mechanism of transport, and the types of transport. To start the topic, I want to give you some introduction about the topic. The fundamental unit of all biological life is the cell. It's a mass of biomolecules in watery solution surrounded by a cell membrane. Cells vary greatly in size, shape, and function, but all cells have common features and components. All the cells have three main regions, namely plasma membrane, cytoplasm that is composed of cytosol and organelles, and the nucleus which has the genetic material of the cell. The cells of multicellular organism exhibit a potential difference across the cell plasma membrane. This potential difference is called the membrane potential, abbreviated as Vm. The value of the Vm is negative. That means the intracellular side of the membrane is negative with respect to the extracellular medium, which is taken as the reference or zero value. The laws of thermodynamics followed by physical and chemical reaction in the living as well as inanimate systems. The second law of thermodynamics suggests that particles with naturally diffuse from a region of high concentration to the region of a lower concentration. The free energy or the potential energy reserved in a concentration gradient can be precisely described. Since free energy is lowest when the distribution of particles is even, the uneven concentration of particles is a milieu with abundant energy. The energy must be incorporated into the system to accomplish the unequal distribution of molecules or to form a concentration gradient. The free energy difference, that is delta G, in moving particles from one compartment with a concentration of C1 to the compartment 2 with the concentration of C2 can be calculated by the following equation, that is delta G is equal to RT ln C2 divided by C1, that is equal to 303 RT log 10 C2 divided by C1, where R is equal to real gas constant, T is equal to temperature in Kelvin. The ability to maintain membrane electrochemical gradient is crucial to life. Electrochemical gradients are generated by the controlled distribution of ions by the transport proteins embedded in the membrane that function as molecular sieve to move electrolytes and solutes across the cell membranes. The active transporter function is driven to ATP hydrolysis. One of the more well-known example of this transporter is the sodium-potassium ATPase, also called sodium-potassium pump, which generates sodium and potassium electrochemical gradients across the plasma membrane. The movement of sodium and potassium ions down their electrochemical gradients can be exploited by the secondary active transporter to energize the flow of other substances through coupled flux mechanisms against their electrochemical gradients. The passive transporter, such as ion channel proteins, provide a pathway for passive movements of ion down their electrochemical gradients. Electrochemical potential. The electrochemical potential is a thermodynamic measure of chemical potential that does not omit the energy contribution of electrostatics. The chemical species, sodium, potassium, ions, water molecules, etc., have an electrochemical potential at any given point in space, which represents how hard or simple it is to add more of that species to that location. If a species will move from region with higher electrochemical potential to an area with lower electrochemical potential, in equilibrium, the electrochemical potential will be constant everywhere for that species. The unit of electrochemical potential is joule per mole. The electrochemical potential is the mechanical work done in bringing 1 mole of an ion from a standard state to a specific concentration and electrical potential. By definition, it is the partial molar Gibbs energy of the substance at the specific electrical potential where the substance is in a specific phase. Electrochemical potential can be represented by the equations given below. Electrochemical potential importance. This is important in biological processes that involve molecular diffusion across cell membranes. Electrochemical potential represent one of the many transferable forms of potential energy through which energy may be preserved. The cell membranes and electrochemical potential are the sum of the chemical potential and the membrane potential. Establishment of membrane potential, how it is established. The difference in the concentration of ions across the plasma membrane determine the membrane potential difference or membrane voltage. Membrane potential in the cells are established by 3 factors. 1. The concentration of ion across the membranes. 2. The permeability of the cell membrane to those ions, that is ion conductance through particular ion channels. And third, by the activity of electrogenic pump, for example sodium-potassium ATPase. The concentration gradient causes the ion to move from the region with the higher ion concentration to the region with the lower ion concentration. The flow of an ion across the membrane that is not balanced by the flow of counter ion leads to charge separation across the membrane, and this charge separation make up the basis for the establishment of a potential difference across the plasma membrane. Charge separation across the membrane leads to the establishment of an electrical gradient that grows in magnitude until it perfectly balances the chemical gradient. Nernst equation. When both the gradients, chemical and electrical, are equal in magnitude, the ion is in state of electrochemical equilibrium, and the transmembrane potential that is created at equilibrium is said to be equilibrium potential, that is VEQ, for that ion under the concentration gradient. It can be given as the equation given below. The above equation is known as a Nernst equation. The Nernst equation permits to estimate the potential that will be established across the membrane based on the valency and the concentration gradient of potassium, provided that only potassium channels are present. This potential is also called as Nernst potential. The Nernst potential for a given ionic species is the membrane potential at which there is no net movement of the ion across the membrane. The ionic species are in equilibrium. Therefore, the Nernst potential of an ion is referred as the equilibrium potential for that ion. At the VQ, the chemical and electrical gradients are equal and opposite in direction. For example, the Nernst potential for potassium ion is the equilibrium potential for potassium ion Vk. The generalized form of the Nernst equation is given below. The electrochemical potential can be calculated by Nernst equation. The Nernst equation describes the equilibrium potential, also known as Nernst potential, for an ion depends on the valency or charge on the ions and its concentration gradient across the cell membrane. The Nernst equation is as follows. VEQ, EQ is at equilibrium, is equal to RT by ZF ln X, concentration of X out divided by concentration of X in, where VQ is the equilibrium potential for a given ion, R is the gas constant, T is the temperature, Z is the valency of ionic species, that is +1 for monovalent cations and +2 for divalent cations. F is the Faraday constant. X out is the concentration of ionic species X in the extracellular medium. X in is the concentration of ion X in the intercellular medium. Calculation of electrochemical potential. The Nernst equation is used to determine the equilibrium potential for various ions. Two examples are given below. First one is for potassium. If potassium ion is the ion of interest, the Nernst equilibrium potential for K, that is Vk, will be, it's given below, where Ki is, or intercellular potassium concentration, is about 150 millivolts and the potassium concentration outside is about 4 millivolts and the valency is 1 because K is monovalent. If the ion of interest is divalent or calcium, if the calcium is one of the ion of interest, the Nernst equilibrium potential for calcium or VCa will be VCa RT plus 2F. Plus 2 is for the valency because it's divalent, ln concentration of calcium outside divided by concentration of calcium inside, where the concentration of intercellular calcium is about 0.000301 millivolts and concentration of calcium outside is about 2.5 millivolts and Z is 2. If there is only one ionic species present in the system and the channels for only one ionic species are open, then VEQ will also be the membrane potential Vm. If there are only potassium ions in the cell membrane, then Vm is in this cell will be equal to the potassium equilibrium potential Vk, which can be calculated by Nernst potential for K. Therefore, in this cell, Vm is equal to Vrest is equal to Vk. When there is more than one type of ion channels that are open at any given time, the situation is slightly different. Now the resting potential can no longer be explained as an equilibrium potential because no ion is at equilibrium, that is Vm is equal to Vrest is not equal to Veq. This is because many ions are contributing to the resting membrane potential, and the ultimate value of the resting potential will depend on the concentration gradients and the relative membrane permeability of the ion present in the system. In this case, Goldman-Hodgkin-Katz or GHK equation is used to calculate the Vm. The equilibrium potential is typically measured in millivolts. The Nernst equilibrium potential values for some ions of physiological importance are given in the table below. These ions of physiological importance are potassium, sodium, calcium, magnesium and chloride, and their potential are given on the right side of the table. Physiological significance of the membrane potential. The normal value of the membrane potential is necessary for many physiological processes. For example, generation of normal action potential in neurons, and influence of the value of the membrane potential on the activity of the electrogenic transport proteins, stimulus secretion coupling in pancreatic beta cells. In order for a neuron to produce an action potential, the membrane potential has to be depolarized or it becomes less negative from the resting value of about around -70 millivolt to the threshold value of around -55 millivolts. Thus, the normal generation of action potential depends on a normal physiological value of the resting membrane potential. The cell membrane. All cells are surrounded by a cell membrane, and this cell membrane is made up of phospholipid molecule, and these phospholipids are fluid and move laterally. There are peripheral protein molecules bound to either side of the membrane, either they bound to inner or outer side of the surface of the membrane. And integral membrane proteins, these are those proteins which permeate the surface of the membrane. The membrane is a fluid mosaic of phospholipids and proteins, and protein can move laterally along the membrane. In this figure, you can show the detailed picture of the cell membrane, phospholipid bilayers, and the integral and peripheral membrane proteins. Transport across the cell membrane. Thermodynamically, the flow of substances from one region to the another can occur in the direction of concentration or electrochemical gradient or against it. If the exchange of molecule takes place in the direction of the gradient, that is, in the direction of decreasing potential, the energy is not required from outside the system. However, if the transport is against the gradient, it will require the metabolic energy. The membrane transport refers to the movement of substance across or through the phospholipid bilayer. Membrane transport depends upon the permeability of the membrane, transmembrane solute concentration, and the size and the charge of the solute. As I told you, plasma membrane is selectively permeable, and there are different types of membrane transport depending upon the characteristics of the substance being transported and the direction of the transport. In this figure, you can see the structure of the lipid bilayer and class of molecules with their examples. Ions and hydrophilic molecule cannot pass through the phospholipid bilayer. Small and hydrophobic molecules can. Mechanisms of transport. There can be different type of transports. First one is the molecule move from the higher concentration to the lower concentration. For this kind of movement, they do not need energy. This is passive transport. It can be facilitated diffusion or it can be simple diffusion. The second type of transport, in which molecular movement against a concentration gradient requires energy, it is kind of active transport. This requires the harnessing of some energy source of the carrier protein. Types of transport, as I told you before, it can be passive or active. Molecules move across membranes by simple diffusion, facilitated diffusion, and active transport. There are three types of transport systems operational to move substance across the cell membrane. Passive, that is simple diffusion, or facilitated diffusion. It is also known as facilitated transport, as you can see in the figure. And the second type is active transport, which require the ATP. The first two mechanisms utilize the power of diffusion and do not require energy. Tremendous differences exist. The rate of transport of substance much higher in facilitated transport as compared to passive transport or simple diffusion. Passive transport. Now coming to passive transport. Passive transport, molecules move down the concentration gradient. As I told you, energy is not required for this process. It may be simple diffusion. Simple diffusion is a passive transport process, which means no energy is required for the movement of molecules. They have natural kinetic energy. Diffusion processes ultimately achieve equal concentration of molecule on either side of the membrane. This is often through semi-permeable membrane. Facilitated transport. It is a form of passive transport mediated by integral membrane proteins embedded within the cellular membrane. Substances enter or leave the cells down their concentration gradient through a specific membrane proteins or channels. These transmembrane proteins form an aqueous pore which allows the ion to move through the cell membranes. Simple diffusion and facilitated diffusion are similar in that no energy is required to move the particles down the concentration gradient. The difference is that for facilitated diffusion, transport protein is required and equilibrium is reached quickly. Simple diffusion allows small nonpolar molecules to pass between the phospholipids to enter or leave the cell in the direction of concentration gradient. Oxygen, carbon dioxide, enter or leave the cell by simple diffusion. Facilitated transport depend molecule size, lipid solubility, and concentration gradient. The transmembrane channels that permit facilitated diffusion can be open or closed. Active transport. Now coming to the active transport, which requires energy. Molecules move across the cell membrane against their concentration gradient from areas of low concentration to a region of high concentration. Since it is an energetically unfavorable reaction, energy is required for this type of movement of molecule, which comes from the ATP hydrolysis. Active transport uses three types of transmembrane proteins, namely uniport, antiport, and symport. Active transport is the pumping of ion or substances through plasma membrane, and it requires a transmembrane transporter protein and energy. The energy source is the hydrolysis of ATP. The energy of ATP hydrolysis may be used directly or indirectly. It can be direct active transport or it can be indirect active transport. It is also known as secondary active transport. Direct active transport. Active transport involves sodium potassium ATPase. The intracellular side of animal cells contain a higher concentration of potassium than that in the extracellular fluid or side. On the other hand, the extracellular fluid contains much higher concentration of sodium than that intracellular side of the cell. These concentration gradients are established by the active transport of both ions by the same transporter. This is called sodium potassium ATPase or sodium potassium pump, which actively transports 3 sodium ions out of the cell for each 2 potassium ions transported into the cell. Sodium potassium ATPase, how it works, the mechanism of action. This pump follows a repeating cycle of steps that consist of the following steps. And the first step is the interior of the pump is open to the inside of the cell, and 3 sodium ions enter the pump and attach to their binding site, and 1 ATP bind to the protein. Mechanism. In the next step, hydrolysis of ATP transfer a phosphate group from itself to the pump, and the interior of the pump is now closed. In next step, the pump opens to the outside and 3 sodium ions are released to the outside of the membrane. Or fourth step, 2 potassium ions from outside can then enter and attach to the binding sites to the sodium potassium ATPase protein. After binding of potassium, this binding causes release of the phosphate group. This causes the pump to change its shape again so that it is again only open to the inside of the cell. The interior of the pump opens to the inside of the cell, and the 2 potassium ions are released. Sodium ions can now enter and bind the pump again so that new cycle can be achieved. Direct active transport. There are few more examples of direct active transport. The proton potassium ATPase. This ATPase is a transmembrane protein. The parietal cells of the stomach use this pump to secrete gastric acids. Another one is the calcium ATPase. A calcium ATPase is located in the plasma membrane of all eukaryotic cells. The energy is provided by ATP hydrolysis to pump 1 calcium ion out of the cell. The fourth type is ABC transporter, or also called ATP binding cassette transporter. They move substances across the plasma membrane. These are transmembrane proteins that expose a ligand binding domain at the one surface and an ATP binding domain at the other surface. Indirect active transport. In some cases, the use of ATP may be indirect. If a cell utilizes ATP to pump out sodium, and then energy stored in the concentration gradient of sodium is used to bring a glucose, the transport of glucose would be an example of indirect active transport. Indirect active transport uses the downhill flow of an ion to pump some other molecule or ion against its gradient. The driving ion is usually sodium, and with its gradient established by the sodium-potassium ATPase or pump, it may be symport or antiport process. Indirect active transport. First one is symport, also called as co-transport. Carrier protein bind two different substrates and transfer them collectively across the plasma membrane. Transport of two solutes is obligatory coupled, for example, sodium glucose transporter. Second one is antiport, also known as exchange diffusion. This antiport carrier protein exchange one solute for another across the plasma membrane. Example, calcium ions are pumped out of the cell by sodium driven antiport pump. Below is the picture. You can see three types of transport. Transport across the cell membrane. In summary, various mechanisms of transport or of ions and molecules across the biological membrane. Simple diffusion, ion or water channel, voltage-gated ion channel, uniport, symport, antiport, and ATP coupled active transport all can be seen in this figure. So, to summarize, the difference in the composition between the intracellular and extracellular fluids depend on the plasma membrane transport system. Because plasma membrane is made up of lipid and proteins, and lipids are making the hydrophobic part of the membrane, and the proteins are the candidate which are making the transport or other molecules to be transported across the cell membrane. The difference in the concentration of ion across the plasma membrane determine the membrane potential difference or the membrane voltage, because we know that the sodium concentration is more outside the cell membrane and potassium concentration is higher intracellularly. And the transport across the plasma membrane can be active or passive. Active transport meaning if the molecule has to be transported, the energy is needed. It is mainly done by the sodium potassium ATPase or sodium potassium pump, in which the pump utilizes one molecule of ATP to transport 3 sodium out of the cell and 3 potassium into the cell. And active transport is thermodynamically uphill in the absence of or against the electrochemical gradient. It can be primary like electrogenic pumps I told you, like sodium potassium ATPase, or secondary by carrier proteins or driven by the electrochemical gradient for one of the substances. So coming to the passive transport can occur by the diffusion mediated or by carrier or channel proteins. The passive transport of ions are largely via channels, because for passive transport there is no need of ATP or energy. It can be simple diffusion, facilitated diffusion, or it may be transport via the voltage-gated ion channel across the membrane.